Enzyme regulation. Enzymes can be inhibited from carrying out their reaction. There are two different types of inhibition that we will mention. One of these is called competitive inhibition. So here's a little simple diagram that represents an enzyme. It has its active site, and so the substrate would fit into that active site. However, there can be other molecules that mimic the substrate, and so their conformation is very similar to the substrate, and they too can fit into the active site, and this would inhibit the reaction. And so it is called competitive inhibition because the inhibitor and the substrate compete for the active site. This is the way that many drugs work. They are designed to fit into the active site, and if the inhibitor molecule is in the active site, that prevents the substrate, and so it will inhibit the reaction. Another type of inhibition is called allosteric inhibition. Allo means other, steric means shape. So here we have an enzyme. Once again, it has an active site. There is a substrate molecule in the active site. But notice on the right-hand side, there is another binding site on the enzyme. If an inhibitor molecule is bound in this other site, it changes the shape of the active site. So this is where the name allosteric comes from. The enzyme now has another shape, and the substrate cannot fit into the active site, and so this would inhibit the reaction. We mentioned earlier that many metabolic pathways involve multiple steps, little small reactions. Each of these reactions has an enzyme that controls that step of the process. And so many times we're going to put these together. So one reaction substrate is turned into a product, and then that product is the substrate for the next reaction. And so we have a chain of reactions in a metabolic pathway. And so here it's illustrated. Here the first enzyme, enzyme 1, and the first substrate, which is reactant A. This is changed into molecule B, and then enzyme 2 changes that into molecule C, and then enzyme 3, and so forth. So a number of reactions that are placed together in this pathway. Eventually we get to end product F. So reactant A becomes end product F, but there are many small steps in the pathway and a number of enzymes. What if we don't need any product F? What if the organism or the cell has all of end product F that it needs? Certainly it does not want to waste energy in all of these small chemical reactions, and so this entire pathway needs to be shut down. This is called feedback inhibition. And so what happens is end product F acts as the allosteric inhibitor for the very first enzyme in the pathway. If we shut down that first enzyme, we're going to shut down the entire pathway. So again, this is called feedback inhibition.